Hello, I'm Udrak Aleton, your facilitator for Bio 216, Chemistry of Carbohydrates, Lipids, and Nucleic Acids. In this video, we'll look at membranes. We'll consider the chemical composition of membranes, we'll look at the functions of membranes, we'll look at the properties of membranes. Please listen attentively and share this video with your classmates. Thank you. So we're looking at membranes. This is Module 2, Unit 5. So at the end of this video, you should be able to discuss the chemical composition of membranes, describe the structure of biological membranes, explain the properties of biological membranes, and enumerate the functions of biological membranes. Membranes, like I would like to describe, are like the walls that make up your room. Assuming your room is a cell, the walls around your room, the boundary of your room are the membranes. The boundary of your fence, the boundary of your room. If you have a container, the boundary or the wall of the container, they are like the membrane. So membranes are like the boundary of your cell. They are the body of I don't okay, let me not call it the body. Let me say just like you have a room, the walls of the room that separates your room from the next room. If you have a big house, the walls that separate your toilet from the kitchen, from the sitting living room, from the bedrooms, those walls are like membranes. And because they separate, the activities that go on in these rooms may not affect the other rooms. For example, assuming you have a house that has good walls, if somebody is using the toilet and another person is in the bedroom, is in the living room, the smell of the excreta will not get to the living room because the door is closed and the walls are well built. So that is what the membrane does to our body. The membrane makes sure that whatever is happening in this cell will not, except it is allowed. Just like I will be in my room, it is only when I open my door that my neighbor will see me. Except it is allowed, it will not affect the other cell of the body. That's what the work the membrane does for us. So we'll look at the chemical composition of membranes. Membranes are composed of two major things: lipids and proteins. We have dealt extensively with lipids in our last video, and most of those lipids we talked about are membrane lipids, or it's called them lipids that are found in membrane. So apart from lipids and proteins, we also have carbohydrates in membrane. So membranes and those carbohydrates in membranes don't occur on their own. For example, you are not going to see starch on your membrane. No, they occur attached to something else. And those things, they are either attached to the protein or the lipids. When carbohydrates are attached to proteins, they are called glycoproteins. When they are attached to lipids, they are called glycolipids. So we have carbohydrates in form of glycoproteins when they are attached to proteins, or glycolipids on the membrane. And then another thing you must note about membrane is that the amount of protein or lipid in a membrane vary based on the activity of that membrane. Let me explain this. In our last video or in our last class, we talked about sphingomyelin. And I made you to understand that this sphingomyelin is found, among other places, in nerve cells. And I explained to you that the work of this sphingomyelin is that it's, among other things, it insulates the nerve cells. Sorry. What do I mean by insulate? When you have an electric wire, you will see that apart from the wire you have inside, they use a rubber coating for the wire. So that rubber coating acts as an insulator, so that when you touch the wire, it does not shock you. That is what sphingomyelin does on nerve cells. Nerve impulses are like electric impulse. So if our bodies are left just without the covering of this fat, this sphingomyelin, anytime something happens, you will receive a shock. And sometimes it happens if you have left your hand, if somebody mistakenly hits your elbow or something, you see that the type of shock you receive. That's what we would have been receiving all over our body if these membranes were not covering our nerve cells. So for such nerve cells, for such cells that have a lot of lipid substances in them, or a lot of lipids in them, their membranes are composed more of lipids. Then we have other types of organelles in the body. For example, the mitochondria. The mitochondria is known as the powerhouse of the cell. A lot of enzymic activities occur in the mitochondria. And because of this, the mitochondria is made up of a lot of enzymes. And due to this, the membrane of the mitochondria is more of protein than lipids. So that's why I said that the amount of protein and lipids in membrane are based on the activity of the membrane. The muscles in our body too contain a lot of proteins, like they have the myosin and the actin. Those are proteins found in the muscle. So we find out that the muscle too has more protein than lipid in the membranes, and different parts of the body. So you can just check through your body now. You have your brain. Your brain has more of lipids in it. So the lipids in the brain the membranes of the brain have more lipid than protein. I don't want us to go further than what I've just explained. So let's the lipids of membrane. The three major lipids components of membrane are the phosphoglycerides. Phosphoglycerides, also known as phosphoacylglycerol. We spoke about them in the last video. Please watch this video, go through your manual, go through your course material, so that these things there will be a continuity for you. These phosphoglycerides, we call them so because they have a glycerol backbone attached to phosphate group and then the fatty acids. Then we have the sphingolipids. Examples of the sphingolipids are the sphingomyelin too that we have spoken about. All these are present in membranes. Then we have the cholesterol. Cholesterol is also a type of lipid present in membrane. So you should know these three major lipids are the major lipids found in membrane. Now for the proteins of membrane, there are two types of proteins found in membrane. Two major types of proteins, sorry. Those are the peripheral proteins and the integral protein. Peripheral, like the word periphery, they are found may be on the surface of the membrane. They can easily peripheral. When you say something is peripheral, that means it's not really attached to the membrane. It can easily be removed from the membrane without hurting the membrane. And then the integral proteins are the proteins that require drastic treatment from the membrane, to be removed from the membrane. Assuming your like I used your wall as what membrane looks like, the integral proteins will be like, they will be part of the block that forms the wall. So if you have to remove them, you have to break the block, and when you break the block, your room is open. So you are destroying the architecture of the membrane. So for integral proteins to remove them, if you remove them, you will disrupt the proteins. And then you also, they are tightly bound to lipids. So we have two types of protein, the peripheral proteins and the integral proteins. And some of the peripheral proteins have enzymic activities too. They are soluble in water. So then the next batch, the carbohydrates of membrane, like that we spoke about, we told you they could either be glycoproteins if they are attached to the proteins, or glycolipids if they are attached to the lipids. So let's write diagram. Then I brought, the membrane is made up of all these things mixed together. That we have in the membrane, we have the lipids, the carbohydrates, the proteins, all cohabiting. So because of the nature of all these things mixed together, this one are all cohabiting, everybody on its own. They call it the fluid mosaic model. And also, the like when we talk about the properties of membrane, you see they say that membranes have fluidity. We'll go back. Lipid, you see, we call it the phospholipid bilayer. You can see it. They arrange themselves in two layers to protect the tail from water, whereas the heads have water. So lipids, phospholipids arrange themselves in this way, and in doing so, they form what we call cells. I mentioned this in our last video when I told you that triacyl glycerols don't form stable micelles. Why don't they form stable micelles? The glycerol has three lipid acids attached to it. There is no free, there is no hydrophilic end again, unlike this one that still has a hydrophilic head. So when you have a under some conditions, the lipids come together and form a sphere, and this sphere is what we call micelle. The hydrophobic ends are away from water, they join themselves inside, and then the hydrophilic end are round, form a round surface protecting the hydrophobic end from water. So this is how micelles are formed. Also, from micelles, like I said in the other slide, they can also form lipid bilayers. They can interact to form two layers of lipid, one layer up, one layer down, and this is called the lipid bilayer. And the polar groups are at the interface between the aqueous medium and the lipid tail. You can see them. The lipid tails are protected from the aqueous medium, and this makes sure that water doesn't get into them. So this formation is known as bilayer formation. And that's because of this formation. These membranes are relatively stable. Apart from this bilayer formation, there's what we at times the bilayer can form a sphere. When it forms a sphere, you can see what we have here. The phospholipids come together and they join themselves and form a sphere. This spherical substance they formed excludes water or whatever, any aqueous solution from the outside and also protects the inside. When this happens, this thing is called a liposome. This spherical vesicle that has been formed is known as liposome. In biology, when you talk about a lot of properties of membrane, we understand the function of these liposomes. So now let's look at the properties of membrane. The first thing we said about membranes is that they have fluidity. And what do we mean by fluidity? We said that membranes are made up of lipids. And when we talked about lipids, we said lipids at times melt depending on the temperature. So those lipids in membranes can also become fluid when temperature is increased, and they can form what will I call it now? They can set too, just like we have lipids, just like what happens to your palm oil, or when you put a lipid in the fridge. At a lower temperature they form. At a higher temperature, they are more they can easily move. So we call this thing fluidity in membrane. Another thing that membranes have is that they have specific recognition sites, also known as receptors. Membranes have specific recognition sites, also known as receptors. These specific recognition sites are the sites, for example, where we have hormone receptors. If a body, if a hormone has a target, maybe a hormone like thyroid now needs to get to your thyroid gland or whatever, there is something in the thyroid gland that when it gets there, it opens the door for it. Beautiful. So these specific recognition sites are like things in the membrane. When you get there, say, okay, let me say they have the key to the door, so they can easily open the door. If you have a door and you don't have the key, you keep on trying, you cannot open it. So membranes have specific recognition sites, so that biomolecules that have those keys can easily open the door and enter into the cell. That's the best way. That's the best analogy I can give you. They also say that membranes, there is asymmetric distribution of lipid components across membrane. What do we mean by this? That the lipid you see inside a membrane may not be the same lipid you will see outside the membrane. They have different types of lipids and they are not arranged in an orderly manner. That's why I said they have asymmetric distribution of lipids across the membrane. Then another thing is that they contain electrically charged surface groups, which supports different electrical potential across the membrane. This also is especially important when you talk about nerve impulse and everything. These electrically charged surfaces allow for impulses and so many other things in the membrane. And then biological membranes allow diffusion of solute molecules through them. I'm sure you remember when you talk about osmosis and diffusion. It's because membranes allow diffusion of substances through them that that it allows you, for example, you are hungry, you eat, your food gets digested and it's in your small intestine. The small intestine membrane opens, allows the diffusion of what you have eaten and have digested to enter into your blood. The glucose in your small intestine can enter into your blood to be transported to give you energy. It is, if not, when you eat, the food will just not have where to go. After the food has been digested, it goes to where it needs. Also, when you take drugs, because membranes allow for solutes to enter, the drug you have taken will go from one place to another. Will go from one part of your body to another. So we can say that membranes have fluidity, they have specific recognition sites, they are asymmetric, there is asymmetric distribution of lipid components, they contain electrically charged surface groups, and the biological membranes also allow diffusion of solutes through them. Next, the functions of membrane. Just like their properties, their functions are are able to occur because of the properties they have. Recognition of signal molecular of recognition of certain molecular signals. Yes, your membrane tell the membranes tell your body, ah, this is this hormone has been put inside to help you do this. This is glue. This drug is here to help you do this. Oh, you have a headache. Okay, all those things. The nerves, the things that happen in your body. For example, the pinch. If I pinch you, it is the nerves, the membrane of your nerves carry the message to different parts of the body. Oh, somebody has pinched the leg, somebody has pinched the hand. So membranes serve as molecular, they recognize molecular signals, they serve as nerve components, just like I've just said. They control movement or translocation of molecules in and out of the body, of the cell. Just like you need glucose, your body takes it. I give you injection, the body accepts it and takes it to where it is needed. They serve as receptors of hormones. These are the properties, the functions. You can see that the functions of membranes are due to the properties they have. Good, let's class activity. What is the name given to the structure formed when a lipid bilayer closes in on itself? Good. Check one of the slides. You will see the answer. So, in summary, we have looked at structures of membrane. We have looked at components and chemical composition of membrane. We have looked at the the lipid bilayer and the mosaic model, the lipid bilayer and the mosaic model of membrane as the most thermodynamically stable. We have looked at the properties of membrane and finally the functions of membrane. Good. Thank you for listening.